My name is Patrick Marks, and I'm presenting the history of the Human Genome Project. So some of the landmarks in genetics and genomics start when Gregor Mendel discovered the laws of genetics, and then the rediscovery of Mendel's work. This is with the pea plants. We learned about this earlier in class. And then, if you read a little bit here, further on, James Watson and Francis Crick described the double helical structure of DNA. Then more things were developed. Stephen Cohen and Herbert Boyer developed recombinant DNA technology. Another thing to notice is Frederick Sanger and Alan Marn and Walter Gilbert developing DNA sequencing methods. Then the GenBank, a database about genomes, was established. And then, even further on, the polymerase chain reaction, PCR, was invented. So these are some of the main things. Finally, as the future goes on in the 80s, there was a lot of talk about sequencing the human genome. And a couple diseases that were genetic were also studied. But during 1990 to the year 2003 was the Human Genome Project. So advances in molecular biology became precedent for the Human Genome Project. The three things that we saw before, the DNA cloning, DNA sequencing, and PCR. And they decided they were going to first sequence yeast, then fruit fly and roundworms, then mice, and then humans. So if you look at this comparison, a human genome and a mouse genome is about 3 billion base pairs, while a fruit fly genome is only 160 million. Nematode roundworm is 100 million, and a yeast is only 15 million. So it'll be easier. Human genome is a lot harder than the ones that they were going to do before. So scientific community's opinions and views diverged about the Human Genome Project. There was no comprehensive step-by-step strategy, and genomics was a growing field evolving from various experts from different fields, and the internet was really not developed yet. So as you see, here's the GenBank. This is all they knew about the genome and what they had in like encyclopedias and their fax machines, no internet. So plans were updated every few years about what they were going to do with the project. So the genomes, first the organisms were organized into chromosomes. Then, see, the human chromosome compared to the human genome is a lot smaller. It's about the same size as a fruit fly or roundworm genome. It's roughly 1 MB is equal to the length of 1 million base pairs. So they take the chromosome, break down into clones, and then make the clones contiguous on top of each other so that they could figure out the sequencing. These then contiguous clones were mapped for the sequencing. The ones inside the red boxes were selected. So shotgun sequencing is a scientific approach utilized to uncover the sequence of an organism's genome or its segments. In this method, the DNA is randomly split into numerous smaller sections, with each section being individually sequenced. Subsequently, a computational algorithm is employed to identify overlapping segments and reassemble the original DNA sequence in proper order. So first they would get the clone, prepare the multiple clones with the PCR, fragment it into subclone fragment. Then with these subclone fragments, they assembled it into the contiguous sequences. With the overlapping sequences, they were able to deduce a rough track sequence, and then they found the final sequence. So here's what like the computer algorithm kind of looked like. When you overlap them, it shows where the spaces are similar. So as more and more get overlapped, you can see a complete sequence. It's shotgun sequencing. So finally, they did the yeast, the roundworm, and the fruit fly. They released it in science magazines. And some of the challenges of sequencing the human genome is that the human genome is 3 billion nucleotide base pairs. The sequence method is in 1990 was about 750 nucleotide base pairs per term. The process needed to be repeated roughly 30 times, and much of the human genome is comprised of DNA that is repeated. So they had automated sequencing and powerful computers to complete these tasks. You remember they had to do it over 30 times. It took 6 different nations, 20 facilities, thousands of scientists. Computed roughly 1,000 base pairs per second, and it was constantly running 24 hours a day and 7 days a week. Now the scientists, they were kind of in a race. Craig Venter and Francis Collins were two different scientists. One was working for like a pharmacy company or like a health company, and one was funded by the government. Francis Collins was funded by the government, and Craig Venter was like for private business. And so they kind of raced to see who would crack the code. Then on June 2000, the rough draft of the human genome was announced, and with these two scientists, spoke with President Clinton to the United States and the world. And then in February 2001, they published the human genome sequence in scientific journals. So some highlights. The Human Genome Project was completed ahead of the 15-year set schedule and under projected budget costs. Human Genome Project was able to produce a sequence of over 90% complete, but still had a few spots missing, so the total cost was about $1 billion. And the Human Genome Project had many applications in biology and medicine. One of the biggest applications that it produced was the use of DNA for criminal profiling. So what about the rest of the human genome? We saw they had the rough draft. The remaining 8% that was undeciphered held significance for structural components like centromeres and telomeres, which had medical relevance. So with the advent of new techniques coupled with enhanced computational strategies, then paved the way for scientists to ultimately produce the full 100% genome. So the entire genome was finally completed in 2022. And this is the article that I did my research on, was this article about scientists who finally, with the new technologies, sequenced the whole entire genome. And this was done just last year, so. And here's my works cited. Thank you very much.